Hi, everyone. Welcome to the 7th Field Notes Live event. My name is Sara Russell, and I am a soil scientist in Gilmer, Texas, and I'm the Field Notes Region 9 representative. And as a regional representative, I serve on the Field Notes Review Committee. As the review committee, we together solicit and select all the topics for each webinar. Today we have two really great topics for you, and we encourage you to ask questions anytime using the Q&A panel. The Q&A panel should open by default, but if for some reason your Q&A panel is not open, simply click on the question mark icon located on the right side of your screen. For closed captions, turn on the live caption button located in the lower right corner. Today's session is being recorded, and the recorded sessions are available in Teams on the Field Notes channel and the National Soil Survey Center's YouTube channel as well. Again, thank you so much for joining us, and I hope that you enjoy today's session. With that, I'd like to turn it over to Dave Hoover to tell you a little more about today's webinar. Dave?
Thank you, Sara. Welcome everybody to the 7th webinar. I think that's really great that we've had great content for 7 of these and looking for at least 7 more. I was at some meetings, some global meetings on soils and spectroscopy a couple weeks ago, and besides debating the heck out of different laboratory methods, one thing that came through is everybody was looking at good field data collection and field activities. And I think this webinar series is a great way of showing our expertise out there by our soils and ecological folks, not only in the foundations of data gathering and data delivery, but in a wide range of other field activities. So I know I've learned a lot just from viewing all these, and I hope you all have too. And I also hope that it spurs you to contact your regional folks for the Field Notes series and suggest a topic of your own so we can hear what's going on in your part of the country. Today we do have 2 presentations, as Sara said, and we'll get into our first one, which is by Brianna Wegner from Fargo, North Dakota, talking about MLRA 53B Closed Depression Digital Mapping Project. Brianna, the floor is yours.
All right, just to double check, is my presentation screen up?
It is.
All right. Okay, we will get started then. Yes, hello, my name is Brianna Wegner. I'm a soil scientist out of the Fargo Soil Survey Office, formerly out of the Bismarck Soil Survey Office, which is actually responsible for MLRA 53B. And I will be sharing with you our closed depression digital mapping project that we have been working on. So the goal of this project was to create a raster soil survey product. And in doing that, we would get a definite sort of value for our accuracy and our uncertainty. And then, of course, update spatial and tabular data, and then also improve our consistency of our map units across county and state lines. And this block diagram here attempts to demonstrate the kind of landscape that we are working with. MLRA 53B is also known as the Missouri Coteau, and it is full of this gentle rolling sloping land. And there really isn't a lot of open drainage in MLRA 53B, so we get a lot of these closed depressions, which also are known as prairie potholes. And so we were focusing just on that part of our landscape as we went along with this project. It was quite a large project. Our scope was over 6 million acres. We had 18 different map units and 8 different target soil series. And this chart here on the slide kind of shows the complex nature of the hydrology that we were dealing with. So we had some recharge areas, some discharge areas, and a lot of sort of interesting hydrology that was associated with some of these landscapes. And so this is just another map for reference, kind of our distribution of our map units. You can see we do have some joint issues that we hope to fix with this project. And just as a little kind of reference note for those of you who might not be familiar with North Dakota, the northern part or the top of this map is the North Dakota-Canadian border, and the bottom part or the southern part of this map is actually north central South Dakota. So we covered quite a large area with this project. And so we started by creating covariates using ArcMap, ArcSIE, and Saga, and these are mainly terrain derivatives. And so there's a list of those there for you to see. And for each of those terrain derivatives, we had multiple different neighborhood sizes that were ran for those. So that's how we ended up with 63 different layers, which we then narrowed down using statistical data reduction using R. And we decided on using the top 4 of those statistically significant covariates to feed into our condition Latin hypercube or CLHS sampling scheme. And those were depression depth, relative position with a neighborhood of 15 cells, Saga wetness index, and convergence. And so those are what we use to generate our sample points. We initially generated 150 sample points, but had to throw one out right away because it was always on a road or in someone's yard, and so it wasn't a really good data point. But then for each sample point that was generated, we had at least one offset point. So this picture in this slide tries to demonstrate that for you guys because it can be kind of confusing. But whenever we would go to a point, it would end in 0, and then we would go up slope, and that pedon would end with a 1, and then we would go down slope and capture that, and that would end with a 2. And so that way we kind of got a nice idea of the different kinds of soils across the landscape. And we were able to get 120 of those central points. And during our 2020 field season, we actually were able to collect 282 pedons, but it wasn't without its challenges. We started our field season the first week of March, which is quite early here in North Dakota. And those of you who maybe live and work in the northern states can agree that it's pretty still inhospitable in March to be out doing field work. But in the September of 2019, we had a lot of rain. And then when October came, it froze. So a lot of these closed depressions that we were planning to work in were filled with water. And we decided that one of the best ways to go about collecting data for this project would be to go out there while the water is still frozen. So we did like the old bog mappers of old did and went out and we chipped through ice, and we decided that took too much effort. So then we upgraded to an electric ice auger and drilled through so that we could get some soils data from some of our soils that maybe are a little bit more wet year round for longer, or that are inundated with water year round and might be a little bit more difficult to get to in the summertime. But things did warm up, and as they did, it brought a new slew of challenges along with it. There was still water in the basins, and so we still had to go out there and figure out how not to get wet. So we put on waders, and then we also brought this little game sled out with us because it would float. And when you sample soils on land, you're able to set your field gear down, you can lay the soil out, and you don't really have to worry about holding anything or anything getting wet. But when you're in the middle of a wetland, it's a different story. So this game sled really helped us. It gave us a spot to put our description kit as well as lay out the soil profile as we were describing it. And then kind of as the summer went on, we moved from having a surplus of water to being in a drought. And we were able to just kind of walk back out to our points yet again. This slide just quick shows you the improvement in our data distribution from when we started, which would be the map with the blue dots. And then the map with the white dots shows where we are at the end of our sampling season as far as our data distribution is concerned. So then once everything was classified and correlated and put in NASIS, we moved on to our model building. We have 401 pedons in our data frame that we then use to create 15 different submodels. And we focus mostly on kind of hydrological properties of our soils that we were looking to model. So things like aquic conditions, whether a soil was hydric or not, you know, ponding duration, drainage class, water depth, but carbonates are also an important part of the story here in North Dakota. So we also submodeled for 2 different carbonate discharge positions that we noticed while we were collecting data. And then once we were pretty satisfied with those submodels, we moved on to knowledge-based modeling. And we used box and whisker violin plots, which are pictured on the slide, to kind of help us give us a starting point so we can see the distribution of our data and know kind of what values we should maybe start with when trying to build knowledgeable constraints for these soils. And we split our targeted soil classes into 2 different sequences, a major depression sequence, which focused on the soils that are actually in the closed depression, and then an upland sequence, which were those soils that show up kind of around the depression or maybe on more of these upland landscape positions around that wetland or closed depression. And then for each of these 2 sequences, we had 7 features of interest that we use. So these will be our submodels that we built in the previous phase, and those are listed there for you guys to look at. And once we got something that we liked, we ended up with a hardened map. And this was a challenge because we quickly found out that things that worked in the northern part of the MLRA didn't always work in the southern part of the MLRA. And so to get something that looked right or looked good across the entire project area was a challenge. And I believe that that is then reflected in our accuracy assessment. So when we tested this, our overall accuracy was 42%. So it's not as great as I was kind of hoping that it would be, you know, but it does show that we kind of had some challenges. And so due to that, we decided that we would use more complexes as opposed to using soil con associations, which is kind of the idea we had at the beginning of this project. But you know, that's the way it shook out. And so this is a quick glance at our error matrix. And I don't really have a whole lot of time to go too deep into it, but what this is telling us is that the further out of the depression we move with our soil model, the less accurate it becomes. And this is mostly because we didn't have a lot of upland soils that were in our training data. And so that explains that for us. So I guess to wrap up some lessons that we learned from this project, it was a really big learning experience, especially for me. And so here's some things that were kind of big takeaways. And the one thing is, don't let the size of an area scare you. You just have to be mindful of some of the challenges that trying to do a DSM project over a large area will entail. So for us with MLRA 53B, we had minor differences in climate as we move north to south. And then also very, very slight variations in the parent material as we move east to west. The till is just a little bit different. The landscape's a little bit different. And then we also encountered a wide range of different landforms or differently shaped landforms that also kind of posed a challenge to us when it came time to do the computer modeling portion of this project. And we also noticed that it was difficult to consistently identify and model our natric areas, and that we will have to do a follow-up project after that to focus on those natric soils. We included 2 kind of in this project just to see if we could do it. But we decided we needed more spectral imagery at the very beginning to help us identify those areas. And then another important thing that we learned was that landscapes should probably be sampled in their entirety for best outcomes. You know, we had a lot of areas that were underrepresented and they didn't really model as well. That was kind of shown in our accuracy assessment and our error matrix. We were kind of hoping that this model would pull out a lot of our closed depressions that maybe exist in upland map units, but it didn't really do that very well because we didn't have a lot of those in our training data set. And then also collecting samples in the late winter or early spring really helped us get a lot of data that we maybe typically wouldn't have gotten if we couldn't have gotten it in the winter time. We probably wouldn't have either walked all the way out onto some of these wetlands in waders, or the water would have been just a little bit too deep for us to get out there. So that was you know, really helpful for this project. And then another thing is that we didn't have just one person straight off the bat to focus on the data once it was in NRCS. And so it got pretty confusing pretty quick in terms of like, who put in this text note, or did we put in a text note, and did it get correlated, did it not? And so I would suggest designating one person to just be the data person, just so you make sure that things are consistent and it doesn't get super complicated super quickly. All right, and so with that, I'd like to thank you guys for taking time out of your day to watch our webinar, and if we have any questions.
Thank you. And we do. So the first question is, did you collect or use any lab data for your training set, or was it just described PET-ON data that you used?
So we didn't collect any lab data, but we had a good amount of lab data from existing PET-ONs and historical PET-ONs from previous update projects that we did use in our training, in our data frame, but we did not actually collect any lab data during the field season.
Okay, thank you. And then I had a question, and I'm not familiar with a model for profile darkness. Could you describe what you mean by profile darkness?
Yeah. Oh, I should have had that out. But so we kind of averaged out the color of our profile to see whether or not it was tended to be more maulic versus non-maulic. And so that is kind of what we meant by profile darkness, is, you know, how dark or close to a maulic color was our profile, and then it was kind of averaged that way as sort of it became a kind of a fuzzy number membership type submodel, in that it really tried to show you where maybe your Pacic soils were or your cumulic soils were, because it would be able to reflect that the darker the soil was kind of corresponded to a number, and then whatever that value was meant that it was dark versus being eric in color. I don't know if I explained that very clearly, but
no, you did. Thank you. The
okay.
concept of fuzzy moloch, I kind of like that one. Another question is, do you plan to get more samples in underrepresented areas to improve the model?
Right now, I think that we are kind of planning to maybe update this model kind of as we go along and do other update projects. And so it's sort of like a jumping off point, if you will. It kind of showed us a lot of things of like what to do differently next time. And so I do believe that in the future, it will get improved, but it'll kind of be as we do other update projects.
Okay, and one last question actually has about 5 questions inside it. We'll do one of them, and then if you could answer the other ones, and they will be kept on record with the broadcast. But were there any issues with landowner contact, and how did you go about handling that?
Right. So yeah, that's one thing I did not mention. We went a route where we contacted the county DCs and asked if they had any landowners who would be cooperative. And then we, called and called again. So we made sure that they knew that we were out there. And so that way we knew that we were working with landowners who, wanted us out there, I guess, in the first place. And then it seemed like every time we would encounter a landowner out in the field that we didn't have any trouble with them. They were like, oh yeah, that's right. Okay, soil surveys out here. So we really didn't have a whole lot of trouble with landowners, but it was kind of tricky in the very beginning to get the county district conservationists to reply to our emails or to, get back to us. But other than that, I think it really helped us find landowners who are, willing to cooperate with us and not really give us any problems. So we really, lucked out and didn't have too many issues with landowners.
Well, you persevered, and glad that you did, and thank you for your presentation.
Thanks.
I want to remind folks that our next webinar is going to be on December 14th. We'll get the agenda out and a reminder email out for that one, but here's an early Christmas present you can give yourself. If you're on vacation, you can gather the family around to watch this one. So please stay tuned next month. Our next presentation comes from Barry Hart from Milan, Tennessee. Going to talk to us about a new chapter for a globally imperiled species, the Harbison's Hawthorn. So Barry, take it away.
Thank you, Dave. I appreciate that so much. I'm hoping that everyone can see my screen.
Perfectly.
Fantastic. Well, folks, this topic is a huge departure from what's typically presented. So I appreciate the opportunity to share what's really more of a story. A few months back, some intrigue was expressed about a very little known and very rare shrub or tree in the rose family. This discussion is centered on that single species, Critiqueus Harvestonia. the Harbison's Hawthorn. I'm beginning with quotations from two publications that were released in 2014, both relating to the rarity of this species. The top quote is from the Guide to the Vascular Plants of Tennessee, and the bottom is an excerpt from the Harbison species account in Hall's, a guide to the hawthorns of the southeastern United States by Ron Lentz. Ron is a leading authority on hawthorns in the Southeast and is a key figure in the story of this species. You'll hear about him later. But it's impossible to discuss this species without first considering the past. The earliest known collection of this hawthorn was made by Augustin Gattinger in 1877, although he did not recognize the plant as something new to science. The most significant contribution was made by Thomas Grant Harbison, for whom the species is named. In 1899, Harbison was in the Nashville, Tennessee area collecting plants for the Vanderbilt's Biltmore Herbarium, which is in western North Carolina. He made a series of collections during the spring, summer, and fall, and his colleague at the Biltmore, Chauncey Beadle, used those to formally describe the species later that year. What's striking about Beadle's account is the statement, numerous examples were observed. After 1916, there were no more records of this species until a second wave of collections were made some 30 years later, but those did not come from the Nashville area. In the late 40s, botanists from the University of Tennessee led an extensive effort to document the flora of the state. Thousands of specimens were collected from across Tennessee and accessioned into the university's herbarium. Based on those efforts, Harbison's hawthorn was discovered in 4 additional Tennessee counties. So based on all observations and collections through 1948, which were not many, this was the known global distribution of the hawthorn, 5 Tennessee counties. This is the same map, but with an overlay of the MLRAs. Based on the information at hand, the 2 most important areas are the Nashville Basin and the southern Mississippi Valley loess. After the 47 and 48 discoveries, no further information was published and no further collections were made of this species. This is both intriguing and shocking because field activities, inventories, and publications on the state's flora increased tremendously from the 1950s through the 90s. This plant seemingly disappeared. In 1982, the Tennessee Natural Heritage Program listed Harbisons as possibly extirpated from the state. The hawthorn was even proposed for federal listing in 1980, but was denied due to insufficient information. Interestingly, in that 1980 Federal Register, the hawthorn was listed as occurring in Alabama, Georgia, and Tennessee. We haven't mentioned occurrences from any other location. There is a reason for this, so hang with me. The numbers at the top of this slide are telling. In Ron's book on haws, he points out that hawthorn authorities in the late 1800s and early 1900s worked independently on publishing descriptions. By 1910, the number of hawthorn taxa exceeded 1,000. And there is a plausible reason for this. Hawthorns are notoriously challenging to identify, which is often attributed to a complicated reproduction. Hybridization, polyploidy, and apomixis have been documented and does occur at some level. These sexual and asexual mechanisms can contribute to variations between populations or even between individuals within a population. So many taxa described during that time period were based on minor differences. By the mid-1900s, however, the pendulum had swung in the opposite direction, and the attempt was to synonymize this massive list of names. So what does this mean for Harbisons? The top statements from the 2 leading authorities really settles it for me. The status of Harbisons as a distinct species has never been in doubt. But there is that question of why this species has been associated with occurrences elsewhere. Well, Harbison's resembles and is related to hawthorns to the south, Ash's hawthorn, which occurs in Alabama and Mississippi, and it has been confused with the three-flower hawthorn, which occurs in Georgia, Alabama, Mississippi, and Louisiana. In 1979, E.L. Little and his checklist of U.S. trees managed to hack the list of hawthorn names down to 35 species. In so doing, Ash's hawthorn was treated as a synonym of Harbison's, and that treatment was carried through the 1990s. So, if one checks the taxonomic status on USDA plants today, they will find that Ash's hawthorn is still treated as a synonym of Harbison's. Now, I'm not picking on USDA plants. I love the service. This is just an artifact of hawthorn taxonomy. But there are literature to support a review of this. Note the map on the left. Those green counties in Tennessee are the actual 1947 and 48 Harbison records we illustrated earlier. I have no explanation why they are treated as three-flower hawthorn. According to the 2014 Guide to the Vascular Plants of Tennessee, Critiqueus triflora is currently not known to occur in Tennessee. Now, for the map on the right, all of the Ash's hawthorn records to the south are treated as Harbison's due to synonymy. This inflates the status of what is a very rare species, which really could affect decision-making regarding its conservation. So recall where we left off earlier that Harbison's had disappeared after 1948. In 1993, Ron Lantz teamed up with the Tennessee Natural Heritage Program to search for it in the Nashville area. In September of that year, they found the species in a park in West Nashville, the Percy Warner Park. After about 1 year of searching, only 2 trees were located, and both were sterile. They did not produce fruit. Ron needed both flowers and fruit to confirm identity, so he grafted material from both trees. His efforts were very successful, and the tree's identity eventually confirmed. They were Harbison's. So the tree had been rediscovered in an area where it had disappeared, apparently since 1916. Altogether, Ron spent 5 years searching Warner Park's at least 20 different additional areas around Nashville and visited the 47 and 48 collection sites with no additional observations. In 1997, one of the original 2 stems that Ron discovered died. With a single tree remaining, it certainly appeared that the species was facing extinction in its natural environment. But the story doesn't end there, at least not yet. In October of 2014, I was conducting reconnaissance surveys on an ESD through the Luss Hills ecoregion in Obion County, Tennessee, with a retired soil scientist, David Thomas, who used to be in our office. Along the edge of a fence row and the rural road that we were on, I noticed a particularly robust shrub that was laden with bright red fruit. Upon a closer look, I could tell it was a hawthorn, but it was unlike any I'd ever seen before. I pondered over the plant for a few minutes, even thought it could have been some kind of crazy escaped exotic, but decided to continue down the road. I left it. After proceeding down that small country road for about 1 mile, I asked Dave to stop the truck for one last look at a forested backslope. As soon as I opened the door, there was a second shrub, just as robust and fruit-laden as the first, and I couldn't turn away any longer. So I grabbed a hawthorn key, and each run through the key kept landing on this exceedingly rare species. So I made the requisite collections for confirmation, and the tentative ID turned out to be correct. I returned to the site the following spring with a former botany professor, Dr. Wayne Chester, along with Ron Lantz. to examine the species in flower and to gather additional information. In all, 36 individuals were counted that day along a 1-mile stretch of road. That's the most individuals of this species that I think had ever been listed in print. And this is the current global distribution of Harbison's hawthorn. Now, one big change on this map compared to the earlier one shown is the removal of one county record from Lawrence County. That record was recently determined to have been an incorrect identification. So it's booted out. The inset topographic map is there to illustrate the dissecting nature of the landscape at the Obion site. And the narrow red polygon represents that road corridor where the species has been observed. So what does this thing look like? The definitive identification of this species requires examination of several characters. The growth habit is one that is generally described as tree-like, but some individuals can take on a shrubby habit, especially if open grown. Under ideal conditions, individuals can attain heights of around 25 feet. The ashes and the similar 3-flower hawthorns to the south tend to be shrubbier and smaller, reaching maximum heights of less than 16 feet. These are a couple examples of fruiting individuals, and note the dense red clusters. That is what initially caught my eye. Individual pomes or fruit usually attain diameters of around 13 to 15 millimeters or about a ½ inch. Thorns are noteworthy. They are stout, mostly straight, lustrous, and range from about 1½ to nearly 3 inches long. They tend to be stouter and slightly longer than the ashes and the 3-flower hawthorns. The leaves are also quite characteristic. They are broadly ovate, oval, or obovate with blades, leaf blades, that range from roughly 1½ to 3½ inches long. I've included a few characters that really stand out to me. The top 3 photos illustrate the petioles of the leaves, the stipules at the base of the leaves, and the calyx, all of which support stalked glands. This characteristic is important to the group of hawthorns in which Harbisons belong, which does include ashes and 3-flower. Now, the bottom photos illustrate the flowering structures, the corm. The number of flowers per corm ranges from around 8 to 12, which exceeds both of those similar to hawthorns to the south. The photo on the left is the rural route where this Harbison population occurs, but the photo on the right illustrates a persistent hazard to individual stems growing along the roadway. The gray dead stem in the photo is one of the original 2 individuals that I found in 2014. This individual had a bush hog set upon it and was later hit with herbicide by the county highway department. So a challenge is to make sure that this population is secured away from the road corridor. One strategy that is currently being pursued along these lines is being spearheaded by the Tennessee Plant Conservation Alliance. And their plan is to propagate seedlings from the Obion site and establish a population elsewhere, preferably on protected lands, probably state lands within the Loess Hills ecoregion. And that's taking a little bit of time and is more easily said than done. But a new opportunity opened up in April of this year involving NRCS. The area biologist for West Tennessee, Mike Hansbrough, and the soil conservationist out of that county, Obion County, Andrea Funk, established a CRP pollinator planting on property where this hawthorn occurs. This was the perfect event and opportunity to finally establish that relationship and connection with the landowner. With Mike and Andrea leading the way, we met with the landowners in May of this year and were welcomed and permissions granted to conduct inventories. Also, another big occurrence this year is over in Nashville. Warner Parks are implementing measures to increase the population there. This is our meeting with the landowners. The gentlemen in the center of the photo have farmed and raised cattle in this area all of their lives. The gentleman over here behind them is their nephew, and Mike, the area biologist, is looking on in the plaid shirt. And words can't express how appreciative I am for Mike and all of his efforts. This is the location of the CRP pollinator planting, which is just coming on in this photo. And it was this practice that really opened the doors for us and potentially this population. With the additional help of the state biologist, Robin Mayberry, and state resource conservationist, Linda Ortiz, I think the Harbison's Hawthorn has indeed entered a new chapter. This is a list of the citations or references that I mentioned throughout the program. And with that, I'll stop and try to entertain or answer any questions. Thank you very much for hanging in there if anyone's left out.
Thank you very much, Barry. Very interesting. There was a comment on excellent observations, but I'm not seeing any questions yet.
Okay.
We'll give a minute here for people who were just typing them in. While we're waiting, have you discussed your findings with the plants database people?
No, I haven't.
His team?
I have not. And at some point, that has to be done. And I really just discovered I've known this plant for a few years now, and I'd never really checked on USDA plants until a few months ago and was very surprised. So at some point, yes, it would be good to discuss this. And there's some great work that's being done on Critiqueus now, which is really wonderful and so badly needed.
Well, Barry, I'm not seeing any more questions come in, but if they do come in, we'll have you answer them, and they will be posted along with the presentations. And I thank you and Brianna for your talks today, for your willingness to share your investigations and your field work with us. I think it was great.
Thank you, Dave.
With that, I'll just remind folks again that the next webinar is set for December 14th. And I will bid all of you a good day.